Cancer cells have a sneaky way of evading the immune system, and much of this trickery lies in tiny messengers called extracellular vesicles, EVs. These microscopic particles can sabotage immunotherapy, a revolutionary treatment designed to help the body's own defenses recognize and destroy cancer. But here's the twist. Scientists are now uncovering how these EVs work their mischief. Thanks to a groundbreaking new profiling method developed by researchers at Purdue University, this discovery could change the game for cancer research and treatment. Led by W. Andy Tao, a professor of biochemistry at Purdue and a member of the Purdue Institute for Cancer Research, the team has introduced an innovative technique that allows scientists to track and identify the proteins and RNA molecules carried by EVs. Their findings, published in the Journal of the American Chemical Society on August 1st, provide a detailed framework for understanding how EVs interfere with the immune system's ability to target cancer cells. The implications? A potential path toward overcoming one of the biggest hurdles in cancer therapy. Extracellular vesicles are like delivery trucks, shuttling a wide variety of cargo, such as RNA, a molecule critical for protein production, from one cell to another. In abnormal conditions, such as in cancer, EVs can transport harmful materials that disrupt normal cellular processes. For example, they can deliver RNA-binding proteins that manipulate immune responses in recipient cells. RNA-binding proteins are crucial because they perform essential functions within cells. Tao explains. Recent discoveries about RNA's expanding roles have fueled efforts to develop RNA-based tools and better understand how RNA-binding proteins operate. The challenge, however, is that studying these interactions has been notoriously difficult. Existing methods lack the capacity to efficiently profile RNA-binding proteins from EVs on a large scale. This limitation has hindered progress in identifying which specific proteins contribute to undermining immunotherapy. That's where Tao's new method comes into play. It addresses this gap head-on. Here's how it works. The researchers attach a synthetic organic molecule to the RNA molecules inside EVs. When exposed to ultraviolet UV light, this molecule creates chemical bonds with any nearby proteins. Essentially, the labeled RNA acts as bait, allowing scientists to fish out the proteins that interact with it once the EVs deliver their cargo to immune cells. By using isotopic labeling, they can distinguish between proteins originating from the recipient immune cells and those already present in the original EVs. Why does this matter? Imagine trying to solve a puzzle with missing pieces. Without a reliable way to separate relevant data from noise, the results would be unreliable. Data science demands high-throughput experiments, Tao emphasizes. However, achieving high throughput isn't enough. Ensuring accuracy is equally vital. If you don't control the false discovery rate, you end up with garbage in, garbage out. To minimize errors, the method incorporates two labeling steps, enhancing precision and reliability. To validate their approach, Tao's team conducted experiments using leukemia-derived Jurkat T cells, a popular model for studying immune responses. They also tested the method on immune cell lines infected with human intrahepatic cholangiocarcinoma, a rare form of liver cancer known for resisting immunotherapy. In both cases, the technique successfully identified numerous RNA-binding proteins transported by EVs, shedding light on how these particles might compromise immune function. One particularly intriguing aspect of this research is its focus on checkpoint proteins, which regulate the immune system's activity. Normally, these proteins prevent the immune system from attacking healthy cells. However, cancer cells exploit this mechanism by sending checkpoint proteins via EVs into immune cells, effectively putting the brakes on the immune response. Could targeting these EV-delivered checkpoint proteins enhance the effectiveness of immunotherapy? It's a tantalizing possibility, and one that researchers are actively exploring. The role of EVs is increasingly recognized in cancer biology, says Tao. His work aligns with Purdue's One Health initiatives, which fosters interdisciplinary research aimed at improving health outcomes across humans, animals, and plants. Supported by grants from the National Science Foundation and the National Institutes of Health. This project underscores the importance of collaboration in tackling complex medical challenges. And this is the part most people miss. While the findings are promising, they raise important questions about the broader implications of EVs in disease. Are we underestimating their impact on other conditions beyond cancer? Could similar mechanisms be at play in autoimmune disorders or infectious diseases? These are just a few of the thought-provoking issues worth considering. So, what do you think? Do you believe EVs hold the key to unlocking the next generation of therapies, or are there ethical concerns we should address before diving deeper into this area? Share your thoughts in the comments below. We'd love to hear from you.